Okay. So our project is pretty different from the one you just heard. We don't focus on any of that. We focus instead on a natural product called podophyllotoxin, and our project is called C-4 analogs of podophyllotoxin as tubulin inhibitors, synthesis, biological evaluation, and structure-activity relationship. So a brief intro into podophyllotoxin. It's a natural product that comes from the plant called the wild mandrake, and this was used traditionally for medicinal purposes by various indigenous cultures, but still holds a degree of anti-cancer value today, mainly because of its ability to inhibit tubulin polymerization and thus prevent the formation of spindle fibers in mitosis, which basically like pull chromosomes apart. So by doing that, podophyllotoxin can inhibit the cell cycle and prevent cell division.
More slowly, please. I can't understand your
Okay.
words when you run over them too fast.
Okay, got it. So you might be thinking, why natural products? Because when you think of medicine, chances are a plant isn't the first thing you think of. But Newman and Craig in 2020 actually report that 2/3 of FDA-approved small molecule therapeutics in the last 40 years are either natural products themselves, derivatized by, or directly inspired by natural products. And within that probability, these synthetic derivatives outnumber natural products themselves in a ratio of 10 to 1, which demonstrates not only the value of harnessing natural products from medicinal chemistry, but also creating alterations on the structure to better target human diseases. So here's a visual representation of podophyllotoxin's mechanism of action. It binds to a pocket within tubulin and thus prevents tubulin polymerization. So as researchers found analogs of podophyllotoxin and evaluated their biological activity, they found that when you change the stereochemistry at C4, which is on blue on your screen right there, and add a sugar, namely creating compounds like etoposide and teniposide, it actually changes the mechanism of action from inhibiting tubulin to inhibiting DNA topoisomerase instead, which established the C4 hydroxyl as a site of biological importance. And there's a lot of like analogs of podophyllotoxin on the web, but our project has a slightly different focus compared to just like finding the most effective or the most potent analog. We actually wanted to resolve a question of how bulk on C4 affects the potency of the compound. So previous studies have not established a clear trend. Some studies found that when you increase C4 bulk, the potency actually decreases, and others found that when you increase C4 bulk, there's no trend. So we wanted to find an answer to that question. And thus we decided to approach our synthesis and evaluation in a systematic manner. Systematic in a sense that if you look on your screen, there's analogs 2A through 2F. And from 2A to 2E, especially, each compound differs just one carbon from the one before and the one after. And we want to evaluate their potency and as well as the mechanism of action in cells and outside of cells.
Yeah, and then another important thing to note is that esters are a metabolic liability. And because of this, we want to see if they were esterase stable in the presence of esterase, which is an enzyme that cleaves esters. So we basically wanted to see if the esters on our analogs would actually cleave off and just leave off podophyllotoxin. And we measured this through the LCMS, where we first created samples of our compounds with pig liver esterase in tris buffer, and then incubated those samples at 37 degrees Celsius for 24 hours. These samples were ran through the LCMS, and then we looked for podophyllotoxin release, which would be evidence for esterase activity. From running the experiment, we saw no podocleavage, meaning that our compounds are most likely stable in esterase. Next slide. And then we also wanted to test the cytotoxicity of our compounds through MTT assays. With MTT assays, we drug cells at different concentrations and then analyze percent cell viability. And the cell viability basically correlates to the amount of cells that are still alive after drugging. And afterwards, we calculate the IC50 values of each of our compounds. And the IC50 values are basically the concentration of drug needed to kill 50% of cells. And a lower IC50 means that the drug is more potent. And from this work, we were able to see that there's increasing IC50s from podophyllotoxin to 4E. And through this, we basically realized that as bulk increases, cytotoxicity of our compounds do decrease, with the exception of phenylacetate. And you can go to the next slide. And then we also wanted to evaluate biological mechanism of action, and this is through cell cycle analysis using flow cytometry. And to give a bit of background information, the cell cycle is the process of like cell division, and with that division of DNA, or sorry, duplication of DNA is done in this process. And our analogs are predicted to inhibit tubulin, so we expect to see inhibition at the G2/M phase where tubulin is present. This is also where there is the most DNA during this process. And then next slide. So we measure our analogs, we measure where our analogs inhibit by drugging our cells with our compounds and propylium iodide. And propylium iodide is a fluorescent DNA stain. And when running the cells through the flow cytometer, we can measure fluorescence and should look for peaks at high fluorescence levels that would indicate high levels of DNA. And then, like I said before, high levels of DNA do correlate to inhibition at the G2/M phase. And that is where we want our analogs to inhibit. And the next slide. And as we can see, our flow cytometry data basically does correlate with our MTT data. You can see with podophyllotoxin and acetate, along with phenylacetate, there is a big peak at G2M. And then with the rest of our analogs, there's just slight amounts of like inhibition that do decrease as bulk increases.
We also sought to use computational docking to analyze how well our esters fit into the docking pocket at the tubulin alpha beta site, and we saw no general trend in binding interactions, which was contrary to our expectations, which was interesting. Next slide, please. So like Brianna mentioned earlier, adding substituents at the C-4 position of podophyllotoxin actually changed the target of the drugs, etoposide and teniposide, to DNA topoisomerase instead of tubulin. So we wanted to see if adding esters at C4 affected the biological target. And to do this, we combined tubulin and GTP with our drugs in buffer and incubated. And then over time, we measured absorbance with the UV-Vis nanodrop, and this allowed us to quantify relative tubulin polymerization. Next slide, please. We use Taxol as our positive control since it is known to promote tubulin polymerization, and we also tested podophyllotoxin to act as a benchmark so we could compare its activity with that of our analogs. Next slide, please. And here you can see that Taxol, which is the yellow line at the very top, had a significantly higher amount of tubulin polymerization, which is as expected. However, our analogs, which lie in between the blue and red lines, they varied a bit in tubulin polymerization inhibition. However, they were roughly within the same range, which was interesting. And small differences include the propanoate ester, which showed the least amount of inhibition, and the pivaloate ester, which showed the most. Next slide, please.
So through our experiments, we found a pretty interesting contrast. So in cell assays, specifically MTT and flow cytometry, we found that the bulkier analogs performed less well compared to less bulky analogs. But then in our docking models, and as Kimberly mentioned before, the tubulin polymerization assays, a lot of these bulky analogs performed just as well or even better than our less bulky analogs. Specifically, if you go to the last slide, Kimberly mentioned how pivaloate, which is one of the bulkier analogs, had the highest percent inhibition of tubulin. So thus we determined that perhaps increasing steric does not directly decrease cytotoxicity. Rather, biological activity may be attributed to the membrane and cytosolic distribution of these compounds. So in any compound, there's an equilibrium between how many are in the membrane and how many are in the cytosol. And increasing the bulkier esters would increase hydrophobicity and thus impact this balance. And we also hypothesized that perhaps biological activity could be attributed to non-specific ligand-protein interactions because there's so many proteins in a cell, we don't know exactly what proteins our compounds are interacting with.
For our future plans, we hope to experiment with adding sugar substituents at C4 to make etoposide-like analogs. And we also want to investigate how altering the structure of podophyllotoxin may impact metabolism in the body. And we hope to do this by introducing an N-methyl group at the D ring and fluorinating at C4. Finally, we would like to thank Edward New, our advisor, for guiding us through this journey, all of the group members who have provided us with so much support, and ASDRP and its sponsors for providing us with this wonderful opportunity to conduct research. And I believe on the next slide, we have a QR to our preprint. Feel free to take a look. Thank you for listening. Are there any questions?
Great job. Great job. I love that data story. That's such a nice.
Wait, let me take a picture of it.